Cysteine lets me talk about some really important concepts in biochemistry, including oxidation and reduction. There's also some fun stuff too, like cysteine is responsible for like curliness of hair and the like snappiness of rubber. And it's all because cysteine can form these things called crosslinks, where basically two of these can hook up. And this can link together like different protein chains or different parts of the same protein. If it happens in a controlled manner, it can be really important for protein function, like holding the chains of antibodies together. But if it happens in an uncontrollable way, this can cause problems because you can be like linking the wrong stuff up. So our bodies have maintain like a reducing environment to prevent that from happening by using like glutathione. And in the labs, we kind of like mimic these reducing conditions using reagents like DTT and beta-mercaptan. And so I just introduced a lot of concepts, so let me break it down.
And these crosslinks, these are like covalent bonds. So these are like strong bonds, not as strong as the ones like the peptide bonds in the backbone and that sort of thing. And they can be like swapped and reduced, as we'll see. But these are not these are different than just like the attractions that can happen between other amino acid side chains. These are actually covalent bonds. And so this is why it's so important that they form correctly and that you don't have unwanted ones form, because then you have these like strong links between different things that you don't want. And so this is why you have to do it in a really controlled way inside of your cells. But these crosslinks allow for these really, because they're these stronger bonds, they're really good at like certifying things to like, especially with like extracellular proteins, so things that like get excreted, things that get like secreted out of the cells. And so outside of the cells, you have a more oxidizing environment. And so those crosslinks can stay put, but inside the cells, you have to kind of make them inside of like special oxidized compartments in the cell, so that they don't get reduced before they can get out into the oxidizing environment. But this can have important functions such as keeping various chains of proteins together through these strong linkages, and allowing them to kind of like survive the journey throughout your body, and not form unwanted crosslinks, but have the wanted ones that are already there. This is the 11th day of 20 days of amino acids, and we're talking about cysteine. And so cysteine is a really cool one. So unlike all of the ones that we've seen before, cysteine actually has a sulfur is right below oxygen on the periodic table. What this means is that they have the same number of valence electrons. So they both have 6 valence electrons. So they can kind of like form the same, they both want to like kind of gain 2 more electrons in order to fill that extra shell. And so they're going to react similarly. And so you'll see a lot of places where you have the oxygen sulfur kind of like sulfur analogs of oxygen-y things. And so like a hydroxyl or an alcohol group would be like an OH, and a sulfhydryl or a thiol would be an SH. And similarly, you can have like an ester, or you can have a thioester where you'd have a sulfur here. And so sulfur could kind of like take the place of oxygen, kind of, they can form like similar types of bonds and stuff, but there are important differences. So because sulfur because it's like a row under that to oxygen, this means it has like a whole nother shell of electrons. And so it has the same number of valence electrons, but they're further from the positive pole of the nucleus. And so sulfur is going to be a less electronegative. So basically it's going to have less of a pull on those electrons. And when it forms a bond to an oxygen, this is going to be oxidizing the sulfur, because the oxygen is going to pull more electrons away from it. One of the things that these cysteine thiol groups is going to let it do is form these crosslinks. So these can be intramolecular, so we're like within the same protein, like 2 regions of the same protein, or they can be intermolecular, so between different proteins. When we have a cysteine, just like on its own, we call it a cysteine, right? And then cysteine, I don't know how you're supposed to pronounce these, but I've just heard cysteine, so that's just like, I guess this would be technically like cysteine, I just say cysteine. But anyway, this is, as we'll see, this is an oxidation reaction. So we're losing electrons. And so these electrons can be going to some sort of, these can be going to like molecular oxygen, which is rare. They can be going to some sort of oxidizing agent. In your cells, this is often happening through like an intermediate, kind of like we'll see like a thiol disulfide exchange reaction like we see with reducing, but this similar thing can be used like in reverse to actually form these bonds in a controlled manner. So when atoms link up to form bonds, they don't always share fairly. So they're forming bonds by sharing pairs of electrons, but they don't always share fairly. So if you have something that's electronegative, it's like electron hogging, like oxygen, it's going to pull the electron density towards it and away from the other things. This is going to make the oxygen partly negative and the other thing partly positive. So if you have an oxidation or a reduction, what this means is that for an oxidation, you're losing electrons or electron density, and for a reduction, you're gaining electrons or electron density. So this is often, we'll see this with, you can think of like if the shared electrons are being pulled away, the thing has been oxidized. So if something bonds to something that's more electronegative, it's being oxidized. So this why it's often accompanied by a bond to forming bonds to oxygen is because oxygen is really electronegative, and so it's going to pull the shared electrons towards it, and so the thing that it's attached to is going to be oxidized. Conversely, if you form a bond to like a hydrogen is not very electronegative, and so then you are basically, it's easier to take the hydrogen's electrons. So they're still shared in this bond, but you're gonna they'll hang out with you more than the hydrogen. And so you'll be gaining electron density. And so we call this being reduced. I like to think of the electrons, the valence electrons, kind of like energetic dogs. And so you can think of these electrons as kind of being, these dogs as being like held in by the leashes, is kind of like your protons. When you have these atoms link up, the dogs can kind of like switch their loyalty. So they might hang out more with one or the other. And so the one that if they hang out more with one of those, we call that one more electronegative. So we can do this thing like calculate like an oxidation number, which is basically saying like, if you were to split these up, who would the electrons share with and what would the charge be on the molecule? So taking into account like how many protons they have and then how many like electrons they would get in this theoretical breakup. But the important thing to know for this post is that basically the oxidation number, you're going to be more oxidized if you were to theoretically split these up, you would get to keep more electrons. And so that's what we're talking about by oxidation. And so we don't actually have to be taking entire electrons. We're not actually like splitting these molecules up. We're just saying if we were to split them up, who would the electrons share with? So if you are bonded to something that's more electronegative than you're going to be losing electrons in that split up. But if you're if you're more electronegative than the thing that you're bound to, then you're going to be getting to keep electrons in the split. Remember, oxidation is loss of electrons, reduction is gain of electrons. So if you're keeping electrons, you're being reduced. So if you're keeping electrons, you're being reduced. If you're losing electrons, you're being oxidized. And so if you're bonding to something like oxygen that's really electronegative, it's going to keep the electrons. If you were to split up, then you're being oxidized. And if you're bonding to something like hydrogen, which you get to keep its electron if you break up, then you are being reduced. So if we look at cysteine, there are like different kind of ways that it can get oxidized. So you have your sulfhydryl, so your thiol group. This can get oxidized, so it can form sulfonic acid, then sulfonic acid, and you add another oxygen, then sulfonic acid, you add another oxygen. And so this is often like an intermediate when you're forming those like unwanted crosslinks, because you don't have that much like molecular oxygen just like hanging around and stuff. But when you have like reactive oxygen species and that sort of thing, you can get these oxidative products, and then they can be kind of like they can serve as intermediates for like sulfonic acid can serve as like an intermediate for these disulfide bond formation. And so, but what we typically talk about when we're talking about cysteine oxidation in biochemistry, we're typically interested in these like sulfur-sulfur crosslinks. So we can see why this is going to be an oxidation, because you're adding bonds to oxygen. And when you're adding bonds to oxygen, what you're doing is the oxygen is going to be pulling those electrons away, remember. And so when the oxygen is pulling those electrons away, you're being oxidized because you're losing electron density. So, and if you go the other direction, you're losing bonds to oxygen. So basically you are gaining electron density because the oxygen is not stealing them from you. But here it's a little harder to see. There's no oxygen involved, but you are swapping out these sulfur hydrogen bonds for sulfur bonds. And so the sulfur is going to be more electron pulley than the hydrogen is. And so you are being oxidized in this process. It's important to note that the loss of the proton, so the loss of, when this hydrogen is lost, it's lost as a proton. And so it's leaving behind the electron. So this is not, deprotonation is not an oxidation reaction. This sulfur has the same oxidation state here and here. It has like the same number of electrons basically like assigned to it. So with oxidation state, it's like, if this were an ionic bond, if you were to split these up, who would the electrons stay with? And so if you were to split this up, the electrons would stay with the sulfur. So if you were to actually split this up, the electrons are staying with the sulfur. And so this, if you take, so this is going to have like the same number of electrons. So we haven't done any oxidation here. We've just removed a proton. If this confuses you, look back to the previous post that I did on redox, and I cover this stuff in detail. So this deprotonation is not an oxidation, but it's an important step on the way to getting oxidized, because it's going to make this sulfur more it's going to make this sulfur more nucleophilic and more reactive. And when we have this deprotonated form, we call it a thiolate anion. So an anion because it has a negative charge. And sulfur is going to be now, especially in this thiolate form, it's going to be very nucleophilic. So a nucleophile is something that like has more electrons that it can comfortably handle. And it's looking for an electrophile, or something that has fewer electrons than it wants and wants to share. And so the reason we call it a nucleophile is because, well, the place where you will find something to help you with your electrons is where the protons are, and the protons are in the nucleus. It's looking for a nucleus that can help these electrons get reined in. And so when you have this thiolate, it's going to act as a strong nucleophile. And so then it will attack an electrophile, and this will allow you to form the crosslinks. So it's a thiolate is actually more nucleophilic than a hydroxylate, so the oxygen version of this. So basically, sulfur is bigger than oxygen. And so what happens is that when you have this in like a watery solvent, the solvent molecules are going to kind of like solvate things. And when you have the sulfur, like, so you're going to have water molecules kind of like surrounding things. And the hydroxylate, because it's smaller, can kind of like get trapped more in a sort of water cage. But this isn't going to happen as much with the cysteine because it's bigger. And so this is going to make it more nucleophilic, so it's going to be able to be better at attacking things. And this is happening, so the solvent effect makes it more nucleophilic, but at the same time, it's a weaker base than serine. So by base, I mean bases when we're talking about kind of like stealing a proton. And so there's more on this in the post that I did on before. There's actually some other differences, like nucleophilicity is a kinetic property, so like how quickly this things happen, and then thermodynamics is like how far, so like at equilibrium, how much of it would be protonated versus deprotonated at a certain pH. And so basically when you act as a base, you're stealing a proton, and the more unstable the deprotonated state, the stronger the base, because the more it'll want to be in the protonated state. And it turns out that the hydroxylate is a lot more basic than the thiolate. So basically the thiolate, it has this bigger electron cloud, and it's kind of like okay with having this kind of like extra electrons. It's got this big cloud that can kind of, you can kind of like spread out the charge more and that sort of thing. But with an oxygen, it's tighter. And so it really doesn't, it's really unhappy like this. And so this isn't a carboxylate. Remember, like we don't have that like resonance, nice resonance stabilizing and stuff. This is just a hydroxyl group. And so this is going to be really unhappy. And so this is going, you're not going to see like serine and stuff just deprotonating the cell. So a thiol is always like, it's not normally going to be deprotonated, but it's not like impossible to deprotonate it, especially like in the context of an enzyme or that sort of thing. It can deprotonate, and this will make it much more reactive. This can happen in an uncontrolled manner, which isn't good, and this can form like crosslinks between unwanted areas of a protein. And so particularly when this happens, this is often happening with some sort of like sulfenic acid intermediate. And why this is that we'll see that when you have one of these reactions, if you just have like two thiol groups, that's not very reactive. A thiol group is going to be more reactive if you deprotonate it. So turn it into a thiolate. So this thiolate group. But even that, it's not going to want to attack a thiol, right? You need to have a better leaving group to leave. A leaving group, basically, when the sulfur is acting as a nucleophile and it's attacking, it's doing this like substitution reaction. So basically, you need something's going to get kicked off in the process. So you're attacking and something else is leaving, your leaving group. So the stronger the leaving group is, the more likely this is actually going to happen, right? Because if this is a really bad leaving group, then this reaction is really unfavorable. But if you have a good leaving group, then it's favorable. And so when you just have an H, that's not a very good leaving group. But if we can put it on something that's like another sulfur-based group that can get kicked off, then that'll allow us to swap things out. And so sulfur swap. And so if this is then attached to like some sort of oxidation product, or if it's attached to another, like a reducing agent, like another thiol reagent, whatever, it has to be attached to something that's going to make there something good to leave, so that this thiol group can act as a nucleophile, attack this, and there can be something something basically attackable, because an SH group isn't really attackable. So this can be kind of confusing if you just look at it like this. But yeah. So anyway, because these crosslinks, you don't want them to form just like willy-nilly and stuff. So in our cells, when we want to form a crosslink in proteins, we typically have those proteins be made like inside like the endoplasmic reticulum, so like the ER, so it's like membrane-bound compartment inside of cells, where we can keep a more oxidizing environment, so that we can keep these crosslinks formed and not have them be broken. Whereas when we're in the cytosol, so when we're in like a general cellular interior, we want to keep a reducing environment. And so we're going to keep a reducing environment using this molecule called glutathione. So it's like this tripeptide that can act as this antioxidant. And so this is going to serve as one of those like thiol exchange agents that we can use to break down cysteine bonds if they form. The glutathione is formed actually from 3 amino acids, so glutamate, cysteine, and glycine. And so this is another way in which your amino acids can play roles other than just being protein letters. In the lab, we typically use things like DTT or beta-mercaptan or TSEP. And basically how a reducing agent usually works is that it's going to, so this is the example of DTT, So you have a thiolate on the reducing agent. It can attack the disulfide bridge. You're going to get like a mixed disulfide intermediate where you have the reducing agent kind of like stuck on one of them. So one of them will get reduced. This one's just swapped for another. But the DTT on this other end, it actually still has a thiolate, so it can attack and form this like ring thing. And so one thing you'll see though is that when you reduce something, you're oxidizing something else. And so we've oxidized the DTT, and now it's kind of useless as a reducing agent. But it has done its job. So when you're using up the glutathione, just as we saw, like the DTT was used up, the glutathione is going to get used up. So it's going to get oxidized in the process of reducing those bonds. And then you're going to have other pathways that can help regenerate, because this is going to be useless as a reducing agent. And so it could actually act as an oxidizing agent, and we don't want that to happen. And so we want to keep a high ratio of this to this. It's a lot easier to control in the lab when we just have to add these like chemicals. Important that we do add these in the lab, so like to our buffers, more like purifying proteins or and that sort of thing, because we want to keep this reducing environment to prevent proteins from forming unwanted crosslinks. So well, also another time this will come into play is when you're doing things like running an SDS page gel to separate proteins by size. Basically, you use a detergent called SDS along with heat to kind of unfold or denature the protein and coat it in this detergent, which is going to give it a negative charge and keep it unfolded but soluble. But the heat and this detergent are not going to break up disulfide bridges. They're just going to like unfold the protein. And so if you want to add, the SDS is going to disrupt all but the covalent bonds. So those cysteine crosslinks are covalent bonds. And so you're going to have to add a reducing agent like BME into your like sample buffer, SDS buffer stuff, in order to break up those crosslinks, or because they'll stay together. But sometimes these crosslinks can have important functions, such as like in antibodies, the crosslinks are actually holding together these different chains of the antibody. Before I end, let me talk more about that hair stuff. So basically, your hair is full of this protein called keratin, and this keratin protein has a lot of cysteines. And these can form these crosslinks between the strands, and this can make the hair kind of curly. And so when you have like a perm, basically what you're doing is you can add a reducing agent to kind of break up the existing disulfide bonds, and then you like wrap it around curlers or whatever. Then you add oxidizing agents to reform them, and now they're going to reform in the place that they are when they're like wrapped around the curlers and stuff. And this is going to be these like permanent crosslinks between the strands. So you're swapping out the crosslinks that were there for like different crosslinks in places that are more desirable for whatever you want your hair to be. And so this is how hair perms work. So you might also be wondering like, well, how about when my hair gets curly when it's like humid? So basically your hair curliness and stuff, it's also going to depend on hydrogen bonding. And so this is not covalent bonds. These are just like interactions, like attractions. And so this is the hydrogen. If you have water, water can break up those, but it's not going to mess with these covalent crosslinks. And so this is why water can kind of make your hair do weird things.